do that. Okay, so Sebastian Mortimer is currently a PhD candidate at Oregon State University in the Department of Botany and Plant Pathology. And Sebastian began his academic career at Willamette University in Salem, Oregon. And after obtaining his BS in biology, he worked on the systematics and evolution of a group of plants called broom rapes, as I understand it. And this work was published in Systematic Biology. And Sebastian then began his MS degree in biology at the University of Idaho. And then after that, he worked as a lab manager at the University of Montana, where he made a departure from plants. He studied mammal coat color change evolution. And then in 2020, Sebastian joined the lab of Dr. Aaron Liston at Oregon State University, where he studies the systematics, polyploidy, and sex chromosome evolution of Fragaria, commonly known as the wild strawberry. Sebastian's research has been published in Science, Genome Biology, and Evolution, NG3. And with that, I will let Sebastian present his talk today, Dimensions of Wild Strawberry Diversity, Polyploidy, and Sex Chromosomes.
Hi, everyone. Thanks for joining us today. Thanks, Robert, for the introduction. Yes, today I'm going to talk a little bit about the research that I've been doing since about 2020, along with Aaron Liston and Tealyn Ashman's lab at the University of Pittsburgh. And I'm going to talk primarily about the genus Fragaria and the diversity that there is in polyploidy and sex chromosomes. And I say diversity a little bit tongue-in-cheek because strawberries are known to be kind of boring florally. They have white petals, and there's 5 of them usually. So it's not really the diverse floral morphology that you get in other groups. For a long time, evolutionary biology has considered the tree of life as a series of branching events. The tips of the tree represent lineages, either still in existence in the present or extinct species from the past. And systematists and taxonomists are often thinking about trying to distinguish monophyletic groups and hierarchical sets that denote common ancestry. But as we've learned more about evolutionary history, biologists have found that the metaphor for evolutionary history is less likely to be a tree than it is to be a braided river delta, where lineages are splitting and then re-merging to form new lineages all the time. Another thing we found is that depending on where we look, we'll see different amounts of branching or merging of lineages. The history of some groups looks to be more or less tree-like, but other groups have a lot more hybridization, gene flow, and reticulation in their past. Also, depending on how far back in time we look, we detect different amounts of lineage splitting and merging. It also matters at what scale we look. Is our view the species level or the population level, or are we looking for patterns at the chromosome or genic scale? I consider myself a systematist, and I find that this graphic here helps me explain the field accurately and succinctly. Systematists are concerned with integrating 3 aspects of biological sciences: taxonomy, phylogeny, and then the processes of evolution. And the most obvious one is the naming of biological groups, taxonomy. And taxonomy itself is a process that can be considered a scientific field, maybe all the way on its own, where we're naming taxonomic units that should serve the communities that use them. Diagnosable taxa, especially species, are critical for biology, conservation, and management. Making informed decisions about taxonomy requires the other 2 pieces of the systematics pie. Using phylogeny to name species is important, and I would argue that more than ever, we're interested in relative amounts of bifurcation and reticulation among biological groups, as well as their current and historical ranges. The last piece of the systematics pie is the processes that are shaping the evolution of groups that we're studying. If you're in the plant sciences, these things are often polyploidy, phenotypic plasticity, and mating systems, or the influence of hybridization, where many researchers are working on secondary contact, gene flow, and allopolyploidy. These three pieces of systematics, taxonomy, phylogeny, and the processes of evolution feed into each other and form one another in a linear way, but also feed back on each other. And that's why I think this figure works well for explaining systematics research. The strawberry genus is an excellent example where all three of the pieces of systematics research are repeatedly used to further the understanding of the biology of these organisms. And the strawberry is also a plant that's famous for its fruit, and which is culturally and culinarily significant. So I'm going to talk today about interspecific gene flow and allopolyploidy. Quick aside about domestication, and then I'm going to talk about mating systems in strawberries. I'm going to start with a historical example that's primarily about people and introduces the main species in my talk. Antoine Duchenne was a teenager when he became interested in botany and particularly in strawberries. His father worked as the king's superintendent of buildings at Versailles, and Duchenne apparently spent his youth playing and then later botanizing in the royal gardens. At age 19, he wrote a natural history of strawberries. This included the first genealogy of plants that included networks between species. A similar genealogy with networks between species, not just a branching pattern, was published a few years earlier and concerned just domesticated dogs. Duchenne's book also had early descriptions of species concepts and descriptions of mating systems among strawberry species. Importantly, he also hypothesized that the cultivated strawberry was derived from a cross between two wild species from the Americas. The book was about 300 pages long, but missing all of the drawings and illustrations that he had done because he didn't have enough money to publish the drawings and illustrations along with the text. For a long time, the lost drawings of Antoine Duchenne remained a little bit of a mystery until another teenager found these. Vivian Lee had been hired by a former US Vice President, Henry Wallace, to research Duchenne for a book on strawberries. A lot of what we know about Duchenne is due to Vivian Lee, who wrote 4 chapters on the history of the strawberry. And through her contribution, we actually have a really nice history of the strawberry. Upon finding the lost drawings, Vivian showed them to Gunter Stout, who was the foremost authority on strawberry taxonomy. Apparently, when Vivian told Stout about the drawings she had found, his face went pale, as he had been searching for them for at least 10 years. It's apparent that Stout could not speak or read French, and Vivian could, so she was able to dig through the library and find them. And this picture of Gunter Stout is what I like to imagine his face was like when he saw those drawings for the first time. The fact is that Duchenne was right about the origin of the cultivated strawberry. It's derived from a cross between Virginiana and Chiloensis. And we actually know a fair amount about strawberries, about these strawberries that led to the original cross. There's good documentation that tells us that 5 plants arrived from South America and Europe. These 5 plants were likely all females. That is, they were male sterile plants that only produce fruit. The North American contribution to the cross was from Fragaria virginiana and had arrived in Europe almost 100 years earlier and was the pollen donor to the original cross producing Ananassa, the cultivated strawberry. As a quick aside, species with separate sexes are rare among angiosperms, and I'd argue that many of them are important ecologically or agriculturally. These include asparagus, hops, aspens, and Palmer amaranth, and of course, strawberries. In the genus Fragaria, separate sex mating systems have arisen several times. We have a range of male and female fertility among individuals. There are strict females, and there are that are male, sterile, and produce no pollen, only fruit. And there are strict males that produce pollen but no fruit. And then there are also hermaphrodites, which produce both. Male sterile morphs can easily be ID'd in the field since they lack anthers. However, differentiating between males and hermaphrodites is difficult. It can only really be done by hand pollination to determine if male fertile plants will produce fruit or not. The two species that are the progenitors to the cultivated strawberry, Fragaria virginiana and Fragaria chiloensis, as well as having separate sex individuals, are also allooctoploids having 4 subgenomes from 4 different diploid progenitors. Each subgenome has 7 pairs of homologous chromosomes, and I've numbered the subgenomes here 1, 2, 3, and 4. We know a little bit about the octoploid strawberry male sterility locus. This is the locus that turns off pollen making in females. It's known from 3 different locations. Location 1 is alpha here in green. Location 2 is beta here in a different subgenome on the same on a copy of chromosome 6. And location 3 is still on a homolog of chromosome 6, but on a different subgenome. Males and hermaphrodites do not have any of this male sterility locus or a sex determining region. And the way that 3 different locations arose is a retrotransposition of a single, the way where the hypothesis about how this arose is that there was a retrotransposition of an autosomal gene, and then 2 subsequent translocations to form 3 different locations. So an individual female has 1 of 3 locations: the alpha in green, the beta location in yellow, and the gamma location in red. The phylogeny of the octoploid SDR recapitulates these transpositions so we can track its movement across the genome. What we know from the initial study is that Fragaria virginiana is polymorphic for the SDR or the male sterility region, but Fragaria chiloensis was only known to contain a single SDR haplotype, and that is the most transposed version, the gamma version. Our first question was then, how is the male sterility gene distributed across strawberry species and the landscape? I'm going to bring back Gunter Stout here, who was the foremost taxonomist on strawberry diversity. He extensively studied diversity of North American strawberry species and described several subspecies of chiloensis and virginiana based on morphology. He also hypothesized that a hybrid taxon had established in the Pacific Northwest, where both Fragaria chiloensis and virginiana are closely allopatric, if not could be considered sympatric. He described morphological intermediates, and since it's widely known that virginiana and chiloensis are interfertile, it's not unreasonable to hypothesize that there are many natural hybrids. Salamone et al. 2013 published the first study to really assess Stout's hypothesis of a widespread hybrid strawberry established in the Pacific Northwest. They sampled broadly across the PNW and found that there were beach strawberries on the coast, which were morphologically and ecologically distinct from virginiana in inland. And they also found ecological intermediates with cytotypes associated with both species. And these ecological intermediates were also morphologically intermediate. This modified our original goal to include an examination of potential hybrid individuals and what sex-determining haplotypes they have. So we're looking at these putative hybrid populations, and they're denoted in pink and as cunifolia in quotes. First, we looked at virginiana, since we know that all 3 SDR haplotypes are present. And this is part of a study that is led by Nevin Colin and Tia Lynn Ashman and the group at University of Pittsburgh, where they were sampled herbarium specimens and collections housed at the USDA National Clonal Germplasm Repository here in Corvallis. We then genotyped the individuals, 220 individuals for the version of the male sterility locus from these herbarium specimens. And we found that there are a number of plants that are female phenotypically, but lack the male sterility locus that we know about. And then we are also able to see that there is a spread, and there's not much of a geographic pattern to the male sterility locus that we know about. Then we wanted to look at the taxonomy in relation to the sex-determining haplotypes. Do we have the current taxonomy of strawberries correct? Is Fragaria chiloensis actually fixed for the gamma haplotype? Is there a naturally occurring hybrid strawberry that is well established in the Pacific Northwest? We gathered whole genome resequencing data from 48 individuals across North America, some from the SRA, and then some newly sequenced accessions. We sampled chiloensis on the coast, putative, intermediates, or putative hybrids that we're calling cunifolia here in pink, and then also Virginiana from inland. We wanted to make sure that we sample across species, populations, and that we examine the nuclear genome, the chloroplast, and the different nuclear sex-determining haplotypes, alpha, beta, and gamma. Using admixture, we found no evidence of a widespread hybrid taxon in the Pacific Northwest. Instead, we found evidence for one hybrid population in the Cascade Range out of the original description of cunifolia by Stout, and those are highlighted here in blue. This result has been independently confirmed by Vance Whitaker's group at the University of Florida, and we think it's enough evidence to sync the morphological intermediates in the Willamette Valley, Coast Range, Puget Trough of the Pacific Northwest into chiloensis. But we also wanted to make sure and take a look at the phylogenetic evidence for this. We estimated a nuclear phylogeny of octoploid strawberries, and here I'm showing a tree for only one chromosome. This is chromosome 1 on the A, or the first subgenome. And this phylogeny has been estimated using 1915 100,000 base paired gene trees from chromosome 1. And those gene trees are then the input for Astral. So what we see here is that their hybrid population from Washington is sister to chiloensis, and that cunifolia is included within the clade, monophyletic clade that is part of chiloensis. Virginiana, we have good support for Virginiana being a monophyletic clade, and that there's some evidence for a separation between east and west. And we see that the SDR haplotypes across the phylogeny in both species. So here I'm showing alpha is found in both species and in the hybrid and in the hybrid population. We find that the beta haplotype, so the first transposition of the SDR haplotype, is found also in both species, and the gamma haplotype is also found in both species. So we've seen a little bit that there's SDR polymorphism across the tree, when we had originally thought that it was maybe that Fragaria chiloensis was fixed for the gamma haplotype. The chloroplast tree shows clustering of chloroplast clades and SDR haplotypes that kind of break our taxonomic boundaries. Here's gamma, which has both species, and this chloroplast clade is somewhat poorly supported, but we still see that we have Virginiana and chiloensis together and a gamma haplotypes. Similarly, in the the beta SDR has both Chiloensis species and Virginiana populations. The alpha haplotype also has both species, the hybrids, and these are spread across the tree. Comparing both trees, we see that the nuclear phylogeny shows a clearer picture of how our phylogeny should or may inform our taxonomy. And we can connect these trees by taxon and color these lines by the SDR haplotypes. So that's alpha, beta, and gamma. And what I find most interesting about this discordance is that among the maternally inherited chloroplast phylogeny and its relationship to the nuclear phylogeny, the SDR haplotypes are somewhat concordant to the chloroplast, and it is the morphologically intermediate populations that tend to have the beta and alpha SDRs, where the strictly coastal populations of Fragaria chiloensis are fixed for the gamma SDR. Just kind of sum up this section, Fragaria chiloensis, there is more polymorphism in the sex-determining locus than we thought. There's little evidence for a widespread hybrid species, but there may be areas of gene flow and hybridization that require further investigation, especially in the North Cascades of Washington. And from this data set, I think we can fairly safely say that we can set aside the idea of ecologically and morphologically intermediate hybrid species in the Pacific Northwest. We also wanted to know more about the effect of the male sterility locus at the population level. For those of you who don't study plants, there's a continuum of mating systems where there are all hermaphrodites on one side. As you move to gynodioece, hermaphrodites and females make up your population. Subdioece, females and males with some hermaphrodites, and then there are strictly dioecious populations with only females and males. Fragaria virginiana and chiloensis probably live somewhere in this realm between gynodioece and dioece. In gynodioecious species, hermaphrodites produce both pollen and seed, and they bear a higher reproductive cost than females. Female frequency is then predicted to increase with increasing environmental stresses in subdioecious populations and dioecious populations, where the females bear the highest physiological demands of reproduction. In these systems, female frequency is predicted to decrease with increasing environmental stress. There's a natural break in the range of strawberries in North America, and this break is more or less along the subspecies delimitations of Fragaria virginiana. And this work, again, led by Nevin Colin, undergraduates at the University of Pittsburgh, and Teal Ashman. Data from this study of herbarium specimens and iNatural's observations shows that Western populations have increasing numbers of females as you move northward, while Eastern populations have fewer females in the north. In the East, populations at higher elevations also tend to have fewer females. In Fragaria chiloensis, we have a slightly different situation. We have one subspecies in North America that's distributed on a single north to south plane for the majority of its range along the Pacific coast. A lot of populations are directly on the beach and only a few yards from the ocean. A lot of the environmental variability, especially in elevation, is reduced compared to virginiana and Fragaria chiloensis. In 2022 and 2023, I went out to survey these beach strawberry populations to see if there's any variability in sex ratio among populations on the Pacific coast. I ended up sampling 18 sites in 4 different states and approximately 226 30-meter transects. And this resulted in 900 or almost 1,000 phenotypes, male or female, and then 400 genotypes, where we took the non-flowering samples or individuals that we encountered on the transects and genotype them for the male sterility locus. The range of flowering sex ratios that we find is 51 to about 10% at each site. The range of non-flowering sex ratio is a female frequency from about 70% to 0%, so no non-flowering females. And what we see is that there's a negative relationship between latitude and female frequency. And the assumption is that Fragaria chiloensis is subdioecious and that northern populations are more stressful for females. And this remains to be tested a lot more. We're also missing a lot of the range of Fragaria chiloensis along international boundaries and the areas that are really hard to get to. And we only know a little bit about how plastic the fruiting traits are among male fertile individuals along this large environmental gradient. One of the pieces of this project that I'm excited to try next is to compare genetic diversity among populations at both ends of the sex ratio spectrum. So sampling these California populations on the left, and then the low female frequency populations from Alaska on the right. As I said in the introduction, evolutionary biologists have found that the metaphor for evolutionary history is likely less tree-like and more like a braided river delta. And one of the best examples of this is allopolyploidy, in which species sharing common ancestry evolve separately for a while and then are reunited through hybridization. And I think this section of the braided river really shows well what allopolyploidy might look like, where you have the river splitting and then coming back together later on. We've always known that there are strawberries in the Oregon Cascade range, but no one had examined them for chromosomal variation. Botanists had assumed that these populations belonged to Fragaria virginiana, but Dr. Hummer at the USDA here in Corvallis noticed slight morphological differences and an entire set of additional chromosomes compared to the octoploid strawberry. So this decaploid strawberry has 5 subgenomes, and it's endemic to the Oregon Cascades. We've sampled, or others have sampled more broadly across the Cascade range and Oregon, looking for the range extent of the decaploid strawberry, and it seems to be restricted to Oregon. The species is also subdioecious, having females and males and a few hermaphrodites. And it's also in Oregon, it's endemic to the higher elevations of the Oregon Cascade Mountains. It has 4 subgenomes that are likely closely related to those of the octoploid strawberry, which is a mix of North American and East Asian diploid lineages. But we have some preliminary evidence based on sequencing read mapping that the 5th subgenome of Fragaria cascadensis is most closely related to a different clade of East Asian diploids. The repeated merging of East Asian and North American diploid species to form allopolyploid species implicates Beringia as a geologic time period that deserves more, or geographic area and geologic time period that deserves a lot more attention if we want to know more about the evolutionary history of polyploid strawberries. What I think is really fascinating about the polyploid strawberries is that polyploids are often gynodioecious, subdioecious, or dioecious, while diploid taxa are for the most part hermaphroditic. And as I mentioned before, Fragaria chiloensis is subdioecious, and the approximate location of the male sterility locus is known. So here I'm showing the octoploid genome with the octoploid SDR locations, all on different copies of chromosome 6. This is the decaploid subgenome genome with a 5th subgenome. I've kind of grayed out the octoploid SDR locations because they don't exist in the decaploid strawberry. But the decaploid SDR location is again on chromosome 6. And it begs the question, what's going on with chromosome 6 and why are there so many male sterility loci on its homologs? And this is going to be a primary focus of my work with our new decaploid strawberry assembly. And this shows where it is in relation to the male sterility locus in the octoploids. The wild strawberries have many more exciting avenues for systematics research. And what we're finding is that there's both deep reticulation between lineages millions to thousands of years ago, but also recent hybridization between extant species and populations. I'd like to thank a lot of lab members at the University of Pittsburgh, Oregon State University, collaborators across the world, and a lot of the field and intel, also the land management agencies, and a lot of the funding that has gone into these projects. With that, I can turn it over for questions, and I'm happy to answer those with the time that we have remaining.
All right, Sebastian, thanks a lot for that great talk. Learning about strawberries, things I didn't know about them. Uh, yeah, if anybody has any questions, you can feel free to ask them or type them out. I'll give everybody a moment to think about it. I'll start out with a simple question. Maybe I missed this, but when you were comparing the chloroplast with the nuclear genome, the phylogenies together, and had the lines matching to indicate the discrepancies. Yeah, exactly. The gray dash, what is that?
Mm-hmm. So the gray dashed species or samples are females that are phenotypically confirmed female, but are missing the SDR that we have described, this translocating gene cassette. And so it's some other mechanism that is causing male sterility in these individuals.
Okay, so you just don't, no one knows.
Yeah, we just don't know what they are yet. But yeah, everything else seems to have these two different, or these three different versions of this ZW chromosome system.
Did you can you scan for a do you think that it's another copy of this type of locus somewhere on the genome that you could scan for, or it could be a completely unique mechanism?
The study in 2018 that was led by Jacob Tennyson found that this is likely not another copy or a different or modified copy of the gene cassette. It's
Mhm.
something else.
Mm.
And it may be a cytoplasmic male sterility locus or situation happening in those species. It's not uncommon in even in the cultivated strawberries for cytoplasmic male sterility to pop up every once in a while. These strawberries maybe seem predisposed to this. Not sure.
Mm. Wow. Yes, Mary Jane,
Um,
Dr. West.
uh, First, I'd like to compliment you on that really interesting history of strawberry systematics. It was especially um, worthwhile, I think. I had kind of a trivial question. I noticed that in your graphs, you were using data from iNaturalist to look at distributions. And I was wondering if you somehow screen for the dependability of the different observations there.
Yeah, thanks for your question. I cannot take credit for the history of the strawberry. My advisor, Aaron Liston, has done a lot of work to put together that history and especially highlighting the work that Vivian Lee did for the book on strawberries. Your question about the iNaturalist observations, and I don't have the map that includes all of the herbarium specimens and the iNaturalist observations, but we went with the research grade. So these are observations that have been confirmed by multiple individuals or multiple iNaturalist users. We also screened a lot of the ones that were kind of iffy ourselves, that are in areas that we weren't quite sure that that they were actually which species they were, because there's can be a little bit of confusion. And then we scored them for male sterility based on Ethan Richardson, an undergraduate at the University of Pittsburgh, screened them for male sterility.
Okay, that reassures me. I think you're overly modest about your interest in history. Not everybody who reads a history appreciates it and puts it in a talk, and I thought you did it beautifully.
Thanks.
And then my other question is, so these different loci, they're like all over the tree. Like there's some pattern, there's some structure to it. Um, but they're in both species, right? They're
Mhm.
in the hybrid. So what is the pattern of evolution to how is that working?
Yeah, at the beginning, I had thought that maybe that in some cases there was some gene flow that was affecting the pattern.
Yeah.
But now I've kind of come around to thinking that the Chiloensis and Virginiana are very young species. The octopoid strawberries are less than 1 million years old, probably, and they have spread across North America, and Chiloensis has even spread to South America and Hawaii. And this has happened really quickly. So what we're probably seeing is incomplete lineage sorting of
Okay.
the chloroplast, as well as um, the sex determining locus.
I see. Yeah, makes sense. Look at those chloroplast branch lengths there.
They're pretty short,
Yeah.
and yeah, that a lot of these populations are very closely related, and I think the species themselves could be they're interfertile. So in some cases, people have considered them that they should be just one species
Hmm.
with maybe different subspecies, just one grade of ecotypes across North America.
I see. All right. Well, if anyone has any questions or thoughts, give you a few more seconds. But if not, thanks Sebastian so much for his talk and for uh, telling us the story of the strawberries here. And we will see you guys next week for the next Greg seminar. Thanks so much for being here.
Thanks, Robert.
Thanks, Sebastian. See you guys.